Hello everyone, welcome to today's webinar. I'm Catherine Dold with CNEN and I'll be moderating today's event. This webinar is titled Biophysical and Structural Biology Methods Enable Fragment-based Ligand Discovery and is being sponsored by WuXi AppTec. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with their mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look for the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected, please log back in according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time through the Q&A box on your screen. Our speaker will answer them at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we are not able to get to in today's broadcast will be answered by email after the event. Please note that CNEN does not endorse any company products or services that may be mentioned in the webinar, and that the webinar will be archived at CNEN Online after the live event. Our presentation today is being sponsored by WuXi AppTec. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable the global pharmaceutical and healthcare industries to advance discoveries and deliver groundbreaking treatments to patients. Through its unique business models, WuXi AppTec's integrated end-to-end services include chemistry drug CRDMO, or Contract Research Development and Manufacturing Organization, biology discovery, preclinical testing and clinical research services, and cell and gene therapies CTDMO, or Contract Testing, Development and Manufacturing Organization, all helping customers improve the productivity of advancing healthcare products through cost-effective and efficient solutions. Our speaker today is Maran Jerebeck-Willemsen, PhD, Head of Biophysics and Screening at WuXi Biology. Dr. Jerebeck-Willemsen studied biology in Dusseldorf, Germany, and did his PhD at Cincinnati Children's Hospital in Cincinnati, Ohio, focusing on the development of gene therapeutic applications involving the MGMT protein and studying DNA damage repair of alkylating agents in hematopoietic stem cells. After receiving his PhD, he joined Nanotemper Technology and was involved in the development of microscale thermophoresis as a powerful technology for studying biomolecular interactions. Since 2017, he has led the Biophysics and Screening Department at WuXi Biology, supporting many clients in establishing challenging biophysical assays and conducting fragment screening using biophysical methods. I'll now hand the program over to our speaker.
In the next 30 minutes, I will introduce you the established workflow and design of conventional and covalent fragment-based drug discovery in WuXi Biology. In addition, I will share the results of 2 case studies from conventional and covalent fragment screenings. Hopefully, after this webinar, you will have a good overview how fragment-based drug discovery enabled the development of new small molecule drugs in WuXi Biology. In WuXi Biology, diverse hit-finding technologies are established, and depending on the project and the target, we will design the appropriate screening strategy together with our partners. High-throughput screening is the method of choice in an in vitro high-throughput biochemical assays available at a relative low protein consumption. DNA-encoded library screening is a very attractive, relatively new hit-finding technology, allowing to screen up to 100 million compounds in a single reaction tube in a short amount of time and with a relatively low protein consumption. New tools and artificial intelligence improved a lot the outcome virtual screening in the last years and can be successfully applied, especially if structural information is available. But today, I will focus on fragment-based drug discovery, which is a very well-established hit-finding technology allowing the development of potent small molecule compounds starting from weak binding fragments to the target protein. Till 2022, we have recorded 58 fragment-derived small molecules which have reached at least clinical phase 1. 6 fragments-derived small molecules are already in the clinic, used as BCR-ABL1, FGFR, KIT, BRAF, BCL2, and KRAS G12C inhibitors. Fragment-based drug discovery is a powerful method to develop a potent small molecule compounds starting from fragments with relatively low molecular weight, low binding affinity, and low structural complexity. The fragments are relatively small and consist of less than 20 atoms. Therefore, a relatively small fragment library of about 3,000 fragments can cover better the chemical space as an HTS library of 1 million compounds. Typically, the hit rate in a fragment screening is relatively high and is between 3 and 10%. A very low hit rate indicates low druggability of the target. The identified fragment hits usually show a relatively low binding affinity to the target protein, but a relatively low complexity of the fragment allows a fast optimization of a fragment to a lead molecule in a relatively short amount of time. Currently, we offer in WuXi Biology a non-covalent and a covalent fragment screening. The non-covalent fragment collection consists of 3,100 fragments, while the covalent fragment collection contains 2,611 fragments with lysine and cysteine reactive warheads. The first step is always to establish a robust binding assay in order to identify the fragments which bind the target. For non-covalent fragment screening, we typically establish MST spectral shift S-pair or thermal shift assays, while the primary screening technology for the covalent molecule is high-throughput rapid-fire mass spec. After the hits are identified and biophysically characterized, we would like to understand how and where the fragment interact with the target protein. Herefore, we would usually use a crystallographic approach via soaking or co-crystallization. Putting together the binding information and the structural information will allow our medchem expert to optimize the fragment and to develop a specific and potent lead molecule. In this slide, we can see some important chemical properties of our fragment collection. The fragment follows the rule of 3, which means that the molecular weight of a fragment is below 300 Dalton, the C log P is below 3, the number of hydrogen-bound donors is below 3, and the number of hydrogen-bound acceptors is below 3 as well. The library is highly diverse and was already used now for more as 3 years in multiple FBDD projects. All fragments were received end of last year by LCMS or NMR and shows a purity above 90%. We usually are willing to share the fragment structures with our partner, so our partner can let their medchem experts have a closer look by themselves on our fragment collection. The success of FBDD project depends a lot on the availability of a robust and fast detection of weak interaction between the fragment and the target protein. In our facility, we typically use S-pair, MST, spectral shift, and DSF for hit ID and characterization. All those technologies have a sufficient high throughput and sensitivity to detect weak fragment binding to the target protein. In S-pair, we can determine binding affinity and binding kinetics of an on-gold surface immobilized target protein. In MST and spectral shift, we can identify binders and measure binding affinities in free solution. In DSF and nano-DSF, we can identify hits affecting protein thermal stability. Every method has its pro and contra, but we think and we made the experience that combining different orthogonal methods help us and increase the success on FBDD projects. During assay setup, we usually establish a binding assay using multiple methods as MST, spectral shift, as well as S-pair and DSF or nano-DSF. Depending on the assay robustness and performance, we would select which technology should be used for primary screening and which of the technologies can or should be used for orthogonal hit validation. The primary screening of 3,000 fragments can be completed in about 1 week. Here on the slide, we can see a typical workflow for fragment-based drug discovery in WuXi Biology. The first step is always the fragment screening, which consists of a binding level screening of the entire fragment library using a single fragment concentration of 0.5 millimolar. The KD of the identified hits will be then determined in the KD ranking measurements. The KD ranking screening allows us to select the best binders. Those hits will be then further orthogonal characterized using another biophysical and, if applicable, a biochemical method. The biophysically and biochemically validated hits will be further studied in X-ray crystallography, either by soaking or by co-crystallography. Here, the goal is to identify where and how the hits bind the target protein. Both the binding affinity data and the collected structural information play an essential role for fragment evolution, which includes compound design and synthesis, binding affinities and kinetics measurement, as well as crystallography. So finally, we attempt to have 2 to 3 series with decent potency and selectivity. Here in WuXi biology, we put many efforts on heat expansion, since we have observed that heat expansion can help to improve the fragment affinities, and by doing so, to increase the crystallability of the fragment hits. This is of course very important, since a successful fragment evolution depends a lot on getting access to structural data. It is much easier to grow and develop a fragment if we know how it binds and where it binds. Here we can see an example of a fully integrated fragment-based drug discovery program consists typically from 2 phases. The first phase includes the fragment ID and structure determination, which include protein, construct design, production, the development of binding assay, the fragment screening, and biophysical hit validation. The hits will be then further characterized in X-ray crystallography. This phase will include heat expansion, of course, as well. After a successful fragment hit validation, we usually initiate the fragment evolution phase in order to improve compound binding affinities and specificity. This phase includes an iterative cycle of compound design, synthesis, and validation of the generated compounds using biophysical, biochemical, and structural biology tools. A fully integrated FBDD program starting protein production of a potent small molecule up to a potent small molecule can take up to 1 to 1.5 years. In the last 3 years, we have performed about 30 fragment screening for our partners using WuXi Biology Fragment Screening Library. All screenings were performed using SPR or MST. Till now, different targets were screened as 3 ligases, transcription factors, helicases, inflammation, and oncology target. Recently, we even screened few RNA as the target molecule. Now I'd like to share with you a fragment screening case study. Here the target was human STING, which is an important endoplasmatic reticulum membrane protein that plays an important role in cancer immunotherapy by activating innate immune response. In order to be able to determine fragments which interact with human STING, we have established MST assay using a physically established positive control called 2,3-CGAMP. The MST assay shows a very good amplitude and good signal-to-noise ratio, which is important in order to detect binding of very small fragments to the target. In addition, the KD determination in MST was correlating nicely with published literature data generated in ITC and SPR. After assay robustness was further optimized, we have run the primary fragment screening using a single fragment concentration of 500 micromolar in duplicates. Here we can see the hits in blue, the non-binder in red, and potential binder in green. In the screening, we have received 8% hit rate. Those hits show, interestingly, either a positive or negative MST response. Since the 2 compounds have shown a negative MST response, it is possible that the fragments with a positive response have a different mode of action. All hits were then further characterized in 12-point KD measurements as duplicates using the Ligand-to-Streak technology. On the right, we can see few nice examples of confirmed binders. To make a long story short, we have screened in total 3,100 fragments against human STING. 4 different biophysical methods were used to validate the hits, including MST, TRIC, non-OTSF, SPR, and NMR. 66 confirmed hits were identified with KD between 10 and 500 micromolar. 9 out of the most promising hits were confirmed in ligand-based NMR. The second part of this talk will be focused on covalent fragment-based drug discovery. Earlier this year, we have launched a new covalent fragment library consist of 2,611 covalent fragments with lysine and cysteine reactive warheads. 42 of the library include acrylamide warheads. All fragments were designed and synthesized in WuXi AppTec. In the next few slides, I will give you an introduction how Covalent FBDD is being performed and executed in WuXi biology. Our highly versatile biophysical toolbox supports an in-depth hit identification and characterization of covalent fragments. For screening, we would usually use a rapid high-throughput mass spectrometry approach. This method allows us to determine a non-reversible covalent binding between the screened fragments and the target protein. Screening of the entire library is performed in single wells, and the primary screening itself can be completed in 2 days. Using nano-DSF, we can further characterize the impact of the hits on protein thermal stability. From our experience, nano-DSF works very well with covalent molecules. Microscale thermophorese or spectral shift allow us to determine an aberrant KD at a certain incubation time. Performing a time course experiment can help us understand the contribution of affinity and reactivity of the tested molecules. Here on this slide, we can see a typical workflow for Covalent FBDD. As expected, the workflow is pretty similar to the previously presented conventional FBDD workflow, but we usually use mass spectrometry method for its identification of covalent molecules, and this method can easily be used to identify covalent binders. The identified hits will be then orthogonal validated, first in nano-DSF, then in MST, and in an in vitro functional assay if applicable. The biophysically validated hits would be further characterized in K-inact over Ki mass spec measurements, which is a very important parameter to assess the performance of a covalent small molecule. Peptide mapping provides an additional information on the binding site. Finally, the biophysically and biochemically validated hits will be further studied in X-ray crystallography, either by soaking or by co-crystallography. Both the binding affinity data and the collected structural information will enable fragment evolution phase. This first covalent FBDD project was performed this year using human BTK. The protein is a key component of B-cell receptor signaling and functions as an important regulator of cell proliferation and cell survival in various B-cell malignancies. BTK is well suited for covalent drug discovery since its kinase domain contains 6 cysteine residues with 2 cysteines on solvent-accessible surface. In addition, multiple covalent positive controls as ibrutinib are available. In order to establish appropriate screening assay condition using mass spectrometry, we have designed tool fragments-based orelabrutinib. In this pre-experiment, 1 micromolar protein was pre-incubated with 5 micromolar compound before running the measurements. As expected, the designed fragment was covalently bound to BTK. Then the assay was successfully transferred to the rapid-fire mass spec device. All fragments were screened as a singletons at a compound concentration of 5 micromolar and protein concentration of 1 micromolar. The screening includes an incubation step of 6 hours. 127 complete binders and 159 partial binders were determined. Complete binders refer to 90 to 100% occupation, while partial binders show occupation between 50 and 90%. Here we can see few examples of the different binders classes in mass spec. In the upper left figure, we see an example for a non-binder. On the upper right figure, we can see an example for a weak binder, while on the bottom left, we can see an example for a partial binder, and on the bottom right figure, we can see an example for a binder which labels BTK completely. Those are the most interesting hits for us. After completing the primary screening, I further characterized the complete binders using different assays. The first assay was nano-DSF, which allows to study the impact of small molecules on protein thermal stability. In this assay, all tested molecules stabilize BTK between 2 to 7 degrees, confirming the mass spec results. Then the most promising stabilizers were tested then in MST, showing an aberrant affinity to BTK in the micromolar range after 30 minutes incubation. In addition, we have studied the aberrant affinity of those confirmed hits without any incubation and after 2 and 6 hour incubation. A 100-fold increase in potency was determined at increasing incubation time. This result suggests a higher activity of the tested fragment that contribute to the potence of the molecule. Finally, we tested those molecules in an in vitro functional assay, showing that the biophysically orthogonal validated hits were active in ADP glow assay, demonstrating that the fragments were able to inhibit BTK kinase activity with IC50 in the moderate nanomolar to low micromolar range. After we were able to rank the most promising compounds, we wanted to study where those fragments bind BTK. Using peptide mapping experiments, we were able to show that the fragment bind and modified cysteine 481 of BTK, which correlates very nicely with our expectations. K-NACT over Ki measurement plays an important role in biophysical characterization of covalent molecules. K-NACT over Ki value describe the relation between binding and reactivity and is crucial for validation of the performance of covalent binders. Here we can see an example of the K-NACT over Ki measurement of one of the most promising fragment hits. Finally, 5 extra co-structures of the most promising fragment hits were collected with resolution of about 1.6 to 1.8 Armstrongs, which is very important for medchem-based fragment evolution. In the future, we possibly will initiate a fragment to lead campaign based on the generated biophysical data and protein fragment co-structures. This will involve multiple cycles of compound design, synthesis, and analytics, with the goal, after a few rounds of optimization, to discover potent and selective BTK inhibitors. Last, I would like to point out our ready-to-go service, including many high-quality assay-grade proteins and relevant biophysical assays, MSD, SPR, nano-DSF, and biochemical assays. Many exciting targets as Werner helicase, KRAS, P53, 3 ligase like Celebron are available. All those targets are fragment screening ready as well. Our fragment screening service can currently be performed either in Germany or in China. I hope you enjoyed the presentation, and I'm glad to answer some questions.
Thank you for a great presentation, Dr. Jarabek. We have a few minutes left for questions. Reminder to our audience, you can submit your questions in the Q&A box at the bottom of your screen, and any questions that we don't get to today will be answered by email after the event. Okay, let's get to our first question. What are the advantages of FBDD compared to other screening methods, and how about compared to HTS?
And that's a very good question, and of course we have the possibility to run different screening of a different technology, but the advantage of FBDD is that we screen a relative molecule with a very low complexity, and therefore we do not need to screen huge libraries, which would be very difficult to do using biophysical screening methods. And the fragments have relatively low complexity, and therefore a library of about 3,000 fragments is completely sufficient in order to cover the chemical space. Another very big advantage that the low complexity allow us to develop the fragment in a relative short amount of time to potent lead molecule where a robust biophysical assay is available in combination with structural biology information. And the technology is very well established now for many years. Regarding HTS, so HTS is the method of choice when we have a robust biochemical assay available, and of course we can just do it in the protein of interest have a function. And again, the advantages is that the low molecular complexity of the fragments allows to cover the chemical space very well.
Okay. For which cases would you recommend running covalent fragment screening?
Exactly. So covalent fragment screening gained a lot of interest in the last few years, and especially if we have a protein where we have a certain amino acid that we want to target, which is typically disease relevant, that of course is a very attractive target for covalent fragment screening. For example, KRAS G12C, KRAS was deemed as undruggable due to very high affinity of the nucleotide to the protein, but a covalent targeting of this of a cysteine on the G12C variant allow us to overcome the very high affinity of the nucleotide to the protein. And it's one example of a protein that everyone considered this as undruggable to make it druggable. We need, but always to keep in mind the drawback of covalent molecules as well, and this is to reach specificity is of course not always very easy, and you always need to balance to look what is the target and what is the most appropriate approach to reach the goal, to reach potency together with specificity.
Okay. And does the covalent library contain components that target other amino acid residues apart from cysteine?
Exactly. So our covalent library collection contains about 40% acrylamide, but we have other warheads to target lysines as well.
Okay. And can you talk about the advantages and disadvantages of covalent FBDD?
Exactly. So we spoke a bit about it. So the advantages again of covalent FBDD is that the reactivity helps to increase the potency of the fragments. The disadvantage again is the nature of the covalent fragment, that it's not always possible to reach high specificity, since it's really important to balance the binding contribution versus the reactivity contribution, since we do not want that our covalent warhead will react with every available cysteine, since many proteins have, of course, cysteine.
Okay. Next question. On average, and from your experience, would covalent fragment hit stabilize or destabilize the protein in DSF-TSA setup?
So typically we see that covalent fragments and covalent molecules in general strongly increase protein thermal stability, so they stabilize it. For the example that we have discussed of the Amgen 510 covalent KRAS inhibitor, it's increased, for example, protein thermal stability over 20 degrees. So we typically want to see, in general, covalent compound, and for non-covalent, it's actually the same, an increase in protein thermal stability. Decrease in protein thermal stability in many cases is an indication for promiscuous binder and a non-specific binder. It's not always the case, but typically we want to avoid working with destabilizing compounds.
Okay. And is covalent binding good with respect to the toxicity of a drug?
Exactly. So I think we have discussed it a bit already. So we always need to keep it in mind that the balance between binding and reactivity, and if the contribution of the binding is big, while the reactivity contribution keep as small as possible, it's possible to reach specificity.
Okay, thank you.
And then, of course, the toxicity will be low as well.
Okay, lots of great questions coming in here. Is FBDD adapted for allosteric binders?
Excuse me, can you repeat the question, please?
Yes. Is FBDD adapted for allosteric binders?
So it can work very well for allosteric binders. For example, if you screen for a kinase, you can you can run the screening in the presence and absence of a binding site compound and just look for an allosteric binder. So you can do it very nicely with many kinases where there are already covalent, there are already very potent binders exist, and then if you will detect binding, that most likely will be a peripheral binder, an allosteric binder. So that's possible.
Thank
and
you.
we did it already successfully in the past.
Okay.
So that's definitely possible.
Next question. How important is structural information for a successful fragment screening project?
From our understanding, extremely important, since it's very difficult to develop a weak binder. And the fragment in the beginning, they are weak binders if you don't know how they bind or where they bind. So the development of a robust soaking system to allow access to structural information is very crucial. So in many cases, we establish and set the characterization system on the same time as we start initiating the screening.
Okay. And how are the fragment library's compounds selected for chemical diversity and solubility?
Exactly. So all our fragments are tested to solubility, and we know that in our 100 millimolar DMSO stock, we do not have any aggregation. We could see the library on regular basis. The entire library was actually QC end of last year, and we always check for solubility as well. In the screening method that we are using, like MST and SPR, we can see if the compound induces protein aggregation as well, and that will treat it on non-preferable hits. And we try, of course, to avoid compounds with low solubility.
Okay. And let's see here. Our next question, when would you typically initiate the structural biology work?
Exactly. So the structural biology work, we would initiate as quick as possible. So that means when we have protein in hand, we would initiate already the structural biology work. And sometimes we would even put an fragment screening on hold if we are not able to develop a structural biology or soakable system. Since, as I mentioned before, the availability of structural information is very crucial to be successful in a fragment in an FBDD project.
Right. Thank you. Let's see here. Next question. What is the timeline of FBDD screening, and what is the average range of hit rate from FBDD screening?
So the timelines for the screening itself, when the protein is available, is about 4 weeks. So we typically need about 1 to 2 weeks to establish the assay, to test assay robustness and reproducibility. When the assay quality is good enough, we initiate the fragment screening at a single concentration. That takes plus minus 1 week, and 1 week more will be to rank the hits depending on their binding affinities, and an orthogonal validation will take 1 more week. So in total, plus minus 4 to 5 weeks is the average time for screening. Hit rate in a fragment screening for the reversible fragments is between 3 and 10%. Significantly low hit rate suggests low druggability. And for the covalent fragment screening, that really depends a lot which protein we are using, and there is a cysteine or lysine which is accessible. And of course, if the protein if the cysteine on a protein is not well accessible, the hit rate for covalent fragment screening will be much lower. So for a covalent fragment screening, that really depends a lot on the target and on the library that we are using. That can vary from 0.5 up to 15%.
Okay, thank you. Next up, how crucial is the conformational state of the protein for FBDD?
Of course, that's very crucial. You know that the protein will be in a conformational state that it can bind the fragment, and especially if you look for a steric binder, you want to screen the protein in a conformation where the protein is can bind the fragments that you are screening.
Okay. And how do you rank FBDD among other technologies to use as the primary screening choice?
Yes, exactly. So we have in WuXi biology, as I presented, different screening technology, and every technology has its drawbacks and advantages. So we see that when there is protein and apoprotein structure available in the public domain, and we can express the protein and produce high protein quality, we think that FBDD have a very big advantage, since we can screen very fast with our biophysical toolbox, and we have a very strong integrated team that can move such a project from protein up to lead optimization very successfully. But of course, if a biochemical assay is available in an HTS format, HTS is another great possibility to develop new drugs. DEL is another very powerful technology that we have a lot of capabilities and skills in WuXi in running. DEL screening is a relatively new technology, but allow us to screen huge amount of on DNA encoded molecule. So we can screen 100 million molecules in a single tube, and that's another a very powerful technique. We see that sometimes combining DEL and fragment screening is a very big advantage in increasing the chance of developing good or potent drugs.
Okay, thank you. A few more questions here. How often do you update the fragment libraries?
That's a great question, since we actually updated our fragment library 2 weeks ago. So actually the library now contains 4,600 fragments. It's still not updated in the presentation, but just to give you an idea, this is now the update number 3 in 4 years. So we update the library on a regular basis. We have a very strong medchem team that always come with new suggestions how to improve the library.
Thank you. Do the STING lead compounds bind at the cyclic dinucleotide binding site? And do you find allosteric binders?
The tool compound, yes. The fragment, we do not know, since we were unfortunately not able, this work was an internal program. We were not able to derive the structures of the fragments, so we cannot say where they bind. And we will need to see if in the future we will put more effort there to try to get the structural information from our fragment hits.
Okay. Is it possible to use SPR or MST for a primary covalent fragment screening?
Mm-hmm. MST, yes, we did it successfully in the past. We have screened 10,000 covalent fragments using MST and did the mass spec after we have finalized the MST screen. That works very well. We are not a big fan using SPR for covalent fragment, since you're going to use a lot of chip, since the compounds are not going to get off the chip, and therefore we would rather use technologies like MST, DSF, nano DSF, or covalent fragment screening if we cannot or do not want to use mass spec approach.
Okay. And which target classes have you successfully worked on in the past using SPR or MST?
So in the biophysics team, we have established in the last 3 to 4 years over 300 different protein targets. So we work on all kinds of assays, a lot of big 3 ligase complexes, helicases, kinases. We even did some work on neurodegenerative targets, RNA targets, and even some membrane proteins and GPCRs. So both technologies are capable to, you can develop assays for each kind of targets, but most important is to get or to purify a protein at the required amount and the required protein quality. So since we are a lot structure biology driven company, we put a lot of efforts on high protein quality. And when the protein quality is high enough, the protein is correctly folded. Typically, we are able to establish an assay.
Great, thank you. Next question. Do you have examples of a fragment window on targets with no known small molecule chemical matter? What would change in your workflow for such targets?
That's an excellent question, and that's and of course an important question. So when we don't have any small molecule to establish this assay, that's of course difficult. What we are doing from time to time is to using cofactors or substrates or even another protein that binds it. Even if you use another protein or an antibody to establish the assay, of course it's not great, but it at least allows us to see that that protein that we use in MST, that the label protein is still functional, and in SPR that we see that the immobilized protein is still okay. And that's, I think, the best what typically we can get. Another approach, what some of our partners do to run, for example, in the beginning, DEL screening, and hopefully to identify potent binders that can be used as a tool compound to establish an assay for fragment screening.
All right. That is about all the time we have today. Dr. Jarabek, do you have any final thoughts you'd like to share with the audience?
No, I just want to thank for the great questions. And if there are any more questions, we will reach me and my colleagues in WuXi, and we will be happy to discuss. We are very excited about fragment screening, and we will be happy to discuss more with you.
Great. Thank you again, Dr. Jarabek, for a fascinating presentation. And thank you to our participants for being such a great audience, and On24 for technology and production services. And thank you, WuXi AppTec, for the sponsorship that made this interactive webcast possible. Be sure to check CNN or CNN Online for information on upcoming webinars. For CNN webinars, I'm Catherine Dold. Goodbye.